Slide 1. My name is Yuki Yoshimatsu, and I work in the field of rare cancer research at the Tochigi Cancer Center. I would like to thank all the doctors involved for giving me the opportunity to present today. In my presentation, I will discuss the sarcoma cell lines that are being established in our laboratory. First, I will provide some background on rare cancers and sarcomas, and then I will talk about the current status and future prospects of cell line establishment. Slide 2. First, let's define what is considered a rare cancer. A rare cancer is defined as a malignant tumor with fewer than 6 cases per 100,000 people per year. There are as many as 180 types of cancers classified as rare. As shown in this pie chart, rare cancers include leukemia, malignant brain tumors, sarcomas, and others. Generally, malignant tumors are named based on the organ where they arise or the gene responsible for the abnormality. For example, lung cancer or liver cancer. Each has its own distinct characteristics. In contrast, rare cancers are grouped together based on statistical frequency, not biological similarity. As a result, there is no common treatment or molecular background among them. For example, leukemia, brain tumors, and sarcomas are completely different diseases, making rare cancer research particularly challenging. Slide 3. Although each rare cancer type affects a small number of people, collectively, they account for a significant portion of all cancer cases. This slide shows the frequency of different malignant tumors. On the left, we see common cancers with large patient populations. As we move to the right, the number of patients decreases. Rare cancers account for about 15% of all cancer cases, more than some major cancers such as stomach or lung cancer. In other words, rare cancers collectively affect a large number of people and deserve focused research attention. Slide 4. This slide shows global statistics. In Asia, countries such as Japan, Taiwan, and Korea show similar trends. About 20% of all cancer patients in these regions are affected by rare cancers. Slide 5. As I mentioned earlier, rare cancers are diverse and have no unifying features, which makes it difficult to define clear research themes. However, clinically, they share a major challenge. The development of effective treatments has been delayed. This delay is mainly due to difficulties in conducting clinical trials and limited pharmaceutical interest because of small market size. These barriers are now being addressed gradually. Slide 6. The core issue is the lack of appropriate models for research. Without these models, progress in developing treatments is severely hampered. Slide 7. In this context, models refers to patient-derived cancer models, which are the main focus of our study group. These include cell lines, spheroids organoids, and xenografts. These models are essential for evaluating anti-cancer drugs and identifying therapeutic targets. Without such models, drug development becomes extremely difficult. The lack of patient-derived cancer models is a major obstacle across all rare cancers. Slide 8. Sarcoma is a rare cancer originating from mesenchymal tissue, characterized by over 50 histological subtypes occurring throughout the body. This diversity, both in histology and anatomical location, makes treatment particularly challenging. Sarcomas often affect young individuals and progress rapidly, highlighting the urgent need for effective therapies. Establishing diverse sarcoma cell lines is essential to better understand the molecular and therapeutic characteristics of each subtype. Slide 9. Sarcomas can arise throughout the body, reflecting their origin in mesenchymal tissues. As shown here, they most commonly occur in the limbs, 25%, but also appear in the trunk, head and neck, retroperitoneum, gastrointestinal tract, skin, and genitourinary organs. This wide anatomical distribution adds to the complexity of diagnosis and treatment. Slide 10. Sarcomas are genetically diverse and often difficult to analyze due to their complex molecular signatures. They include both bone and soft tissue sarcomas with a wide histological spectrum. Fusion genes are found in about 1/3 of sarcomas and are often subtype specific, offering important diagnostic and therapeutic clues. In addition to gene fusions, sarcomas exhibit distinct DNA methylation profiles and gene expression patterns. further reflecting their molecular heterogeneity. Slide 11. As shown on the left, the 5-year overall survival rate for osteosarcoma improved significantly in the 1970s. However, since then, there has been very little progress. Despite ongoing research efforts, sarcoma grades and survival rates have remained largely unchanged over the past few decades. Currently, there are only 9 types of anti-cancer drugs that are effective against bone and soft tissue sarcomas, as shown in the timeline on the right. These include targeted therapies and immunotherapies, but the development pipeline remains limited compared to more common cancers. Slide 12. Currently, we face a major limitation: the lack of reliable experimental models that allow for effective proteome analysis. Without these models, it is extremely difficult to identify biomarkers that could guide diagnosis, prognosis, or therapy. This slide shows several techniques used in proteomic research, such as 2D-DIGE and reverse phase protein arrays. These methods are powerful, but they rely on well-characterized models to be meaningful. Therefore, creating robust models is a crucial step if we want to discover clinically useful biomarkers and ultimately improve outcomes for patients with sarcoma. Slide 13. This slide summarizes the process of biomarker development. After identifying candidate biomarkers, we perform functional analyses. Verification experiments are a critical step in biomarker development, but they require the cooperation of many researchers and clinicians to collect clinical specimens for validation. In the case of rare cancers, it is uncommon to have a sufficient number of clinical specimens from the beginning. Therefore, we aim to demonstrate that the candidates are functionally relevant biomarkers and use that data to engage with and gain support from clinicians for further collaboration. To clarify the functional roles of these biomarkers, patient-derived cancer models, such as cell lines, organoids, and xenografts, are essential. Slide 14. Research often begins with clinical observations, such as metastasis, recurrence, or treatment resistance. At the same time, hypotheses may arise from laboratory studies of molecular abnormalities. We believe that patient-derived cancer models serve as a bridge between clinical observations and laboratory hypotheses. Slide 15. In response to these challenges, we developed patient-derived cancer models, including cell lines, spheroid organoids, and xenografts derived from tumor tissue. Slide 16. This slide shows a screenshot from Cellosaurus. It provides information on the number of registered cell lines and summarizes them from various perspectives. Slide 17. We investigated how many sarcoma cell lines have been reported among the cell lines registered in Cellosaurus. To do this, we checked each WHO-classified sarcoma histological type to see how many relevant cell lines existed. Although Cellosaurus has a search function, it was not sufficient for our needs. So, we downloaded the full dataset from the area indicated by the red line on this slide and analyzed it using Python. Slide 18. This slide shows the process of systematic review, are registered in Cellosaurus. If you narrow down to Homo sapiens, you will get 97,000 strains, and if you narrow down to sarcoma, you will get 1,728 strains. Excluding the cell lines created by introducing genes into specific cell lines, the number of sarcoma cell lines was 1,013. This slide summarizes how 1,013 cell lines are reported and stored. According to the survey, there are 149 cell lines stored in the cell bank and 923 cells published in the paper. The 121 strains shown in red are easy to use cell lines because they are also in the bank when actually used for research and there is information in the paper. 62 strains are cell lines that are not stored in cell banks and have not been published in papers. Such stocks are virtually difficult to use. Slide 19. This slide summarizes the breakdown of sarcoma cell lines registered in Cellosaurus. What type of sarcoma tissue is the cell line established for? This slide is an excerpt of the top 20 that summarizes the number of established cell lines by histological type. 100 cell lines have been established for top 3 osteosarcoma, Ewing's sarcoma, and rhabdomyosarcoma. It is not clear why cell lines are often established for these three sarcomas. It is not the most common sarcoma, and there are other sarcomas as malignant as these three. Slide 20. This slide summarizes the frequency of pediatric malignancies extracted from the SEER, Surveillance Epidemiology and End Results American Cancer Registry System. SEER is a cancer statistics database that covers about 26% of the American population. SEER data show that osteosarcoma, Ewing's sarcoma, and rhabdomyosarcoma are the most common pediatric sarcomas. Top 3, which has many established cell lines, one possibility is that osteosarcoma, Ewing's sarcoma, and rhabdomyosarcoma are also pediatric sarcomas. And sarcoma research in the pediatric field is more active than adult sarcoma research. There may be, and that may contribute to the high number of cell lines. Slide 21. To summarize the results so far. First, a large number of cell lines were established and reported for a very small number of histological types. Regarding why the establishment of cell lines is concentrated in some sarcomas, the tendency of established and reported sarcomas does not seem to be related to the frequency of occurrence and the number of cases. That has not been established even if the malignancy is high, so it seems that it has nothing to do with the malignancy. The reason why cell lines of osteosarcoma, Ewing's sarcoma, and rhabdomyosarcoma are well established may be that they are pediatric sarcomas, which may indicate the activity of the research field. Maybe. Slide 22. In such a situation, such measures can be considered from the standpoint of establishing a cell line. First, the policy is to concentrate on sarcoma tissue types for which cell lines have never been established, unless it is a cell line that has not been established because it is difficult to establish, I think. Second, it is possible to focus on sarcoma tissue types for which no cure has been established. Since it is a cell line that is indispensable for drug efficacy evaluation tests of anti-cancer drugs, we would like to prioritize the establishment of sarcomas for which effective anti-cancer drug treatment has not been established. But there are many sarcomas for which no treatment method has been established, so it can be difficult to target with this policy. Another option is to establish a cell line from the sarcoma tissue type for which tumor tissue is available. There are few sarcomas for which a sufficient number of cell lines have been established, and since any sarcoma is important for patients, I think there is an idea to work on what tumor tissue is available. With this method, you can concentrate your efforts where you have resources, so it is an efficient way to proceed with experiments. Cell lines that researchers I know are looking for. Since cell lines are a meaningful resource only after they are used, I think it is a good idea to create a cell line that has already been confirmed by the user. If you proceed with this method, you may want to conduct a questionnaire survey or a literature survey to find out the demand. As far as I can tell, there is a shortage of cell lines for most sarcomas, so it is possible that there is no research and there is no demand because there is no cell line. So just because there is no demand, I don't think it's necessary to make a cell line. About it. Slide 23. As I mentioned in the previous slide, there is a vicious cycle in which research is not conducted without cancer models such as cell lines, and there is no demand for cancer models. In vitro studies are rarely available. We believe that such a vicious circle of rare cancer research due to the lack of a cancer model exists in various rare cancers. Slide 24. Cancer model becomes available for such a situation, we believe that functional analysis will be performed to answer clinical questions. Then, the idea of a breakthrough treatment method by new biology will come up, and it is expected that the research results will be returned to clinical and social situations. Based on this idea, we are creating cell lines for rare cancers and sarcomas. Slide 25. Using the established sarcoma cell line, we are conducting drug efficacy evaluation tests for anti-cancer drugs and conducting proteogenomics analysis in parallel. When there is a mutation in a gene that is considered to be a therapeutic target, is the corresponding molecular target drug really effective? And what is the molecular background of the efficacy of the molecular target drug that shows anti-tumor effect without the mutation? We are investigating whether there is a biomarker for predicting efficacy at that time. Slide 26. This slide is an example, but a drug efficacy evaluation test was conducted using an established cell line. The heat map shows the tumor cell survival rate when each anti-cancer drug is administered. In this group, the group on the left, the part surrounded by red, shows the effective drug group. Some of the most effective anti-cancer drugs were those that suppress the signal transduction system, those that suppress the epigenomics pathway, and those that inhibit the proteolytic pathway. Slide 27. Molecular level analysis, NCC oncopanel analysis, was performed to identify mutations in the target gene. We also identified treatment-related mutations from SNP array analysis. Mass spectrometry also identifies molecular pathways that can be therapeutic targets. We are in the process of integrating such data and conducting research toward the development of new treatments for sarcoma. Slide 28. By integrating various types of molecular data, we hope to better understand rare cancers and move forward with more effective, personalized treatment approaches. Slide 29. That's it. Thank you for your attention. I would like to express my sincere gratitude to all the researchers who contributed to this work. It was a great pleasure to share our findings with you.